Alright, so this is my video for DNA replication, and I have the 8 steps here, and we'll go through the whole list here and explain everything. So step 1 is the gyrase. What the gyrase does, it helps to relieve the tension between these ones. And then once it does that, step 2 comes into place, which is helicase, which these are both enzymes because they have ACE on them. So number 2 is helicase. Which helicase does, helicase cuts down the hydrogen bonds, which you can see here in the little dots in between. It cuts down the hydrogen bonds and separates the DNA in half. And then, on the third step comes in the single-strand binding proteins, which is otherwise known as SSBP. And what that does is, the single-strand binding proteins attach to the halves and keep the DNA molecules separated. Right? And which, obviously, they are needed because the sides are attached to each other, and without the single-strand binding proteins, they would move back together, which helps them separate here. And then number 4 are the lead and lagging. And what the lead and lagging do, it's for the replication fork, which it forms a lead and lagging strands, which comes in through over on this part, through here. And then number 5, the RNA primase, which is also an enzyme because it ends in the ACE. And what the RNA primase does is, in the leading strand, which comes back to this step 4, in the leading strand, the RNA primase moves along nucleotides and coats with an RNA primer that will be used as like a homing beacon, which you can call it, it tracks the other thing, a homing beacon for the DNA polymerase. So it tells the DNA polymerase where to start on here. And then, on to the next one, which is DNA polymerase, which starts with number 3, not 1, which is sort of weird. Has DNA polymerase 3, and what it does there is it attaches a post-primase to DNA nucleotides and moves them along towards the DNA replication in the prime 5 and 3, prime 5 and 3 down here, and then there's a prime 5 and 3 down here. Moves them across all the way there, and then it adds the nucleotides to the half DNA molecules through here. And then on to step 4, DNA polymerase 1, which is just the first of the third, and it's still an enzyme because it has the ending ACE in it. And what that does is the other polymerase 1 replaces the primer with DNA nucleotides, so all the DNA is actually there. And then on the last one, what ligase does, it's the 8th step. Ligase does, it attaches up all the gaps, which comes in through here, and all the spaces in here. And this is just showing you which ones are which, the thymine, adenine, guanine, cytosine, which are all the bonded through there with the hydrogen bonds. And then you have your deoxyribose sugar, which is on the outsides, and then the phosphates through here, each nucleotide. And those are basically all the steps of DNA replication, broken down into 8 parts.